Today we will be discussing about reproduction in bacteria. So why bacterial reproduction is important is because you will learn that these helps in the transfer of genes. So generally when we talk about bacteria, it just contains a circular DNA. Now in some cases you might find extracircular bodies, and these extracircular bodies are called plasmids or the F factor. F factor and plasmids are the extra chromosomal or genetic material in bacteria, and in some cases within the bacterial chromosome itself a small portion of the gene means HFR. HFR stands for high frequency recombination of bacteria. So when we talk about bacterial reproduction, the first thing we have to know is about bacterial DNA. That means it is a circular material, and in some cases it contains the HFR. HFR stands for high frequency recombination, and in most of the cases you will find extra chromosomal material which is either the plasmid or the F factor. Now because of this F factor, if a bacterial cell has F factor, it is designated as F plus cell. If the F factor is lacking, it is designated as F minus. Now why your F factor is important is it helps in the transfer of the genes from one cell to another. So basically the presence of an F factor in a cell, that bacterial cell is known as the main. So how this F factor works is by conjugation. By conjugation, what it means is genetic transfer takes place due to contact between two bacterial cells. Generally, the contact between two cells is such that one bacteria has the F factor, so hence this is an F plus cell, and the other does not have any F factor, so this becomes your F minus cell. So these two are designated as male and the female. F plus cells, they always avoid F plus cells. So what happens is this F factor is responsible for forming a cytoplasmic bridge. The genes present in the F factor forms a cytoplasmic bridge. So what happens is DNA replicates, the F factor also replicates, and a portion is transported to the F minus cell. Over here, it completes its replication, and this cell is now converted into an F plus cell. The moment it is done, the cytoplasmic bridges are broken off, and the gene transfer has already taken place. So this is a method of reproduction in bacteria, right? Conjugation, in which gene transfer has taken place. Now why a conjugation is important is, if you remember in your earlier chapter when we were discussing DNA as a genetic material, there were many such experiments like transformation. So in order to understand this conjugation was discovered by Lederberg and Tatum. They observed this in a bacteria called Escherichia coli. So these bacteria have the ability to synthesize their own growing materials which are necessary for their growth. So let's say two strains of E. coli are capable of growing in different mediums. In this particular medium, A and B growth factors are present and C and D growth factors are absent. In this particular medium, growth factors A and B are absent and growth factors C and D are present. That means bacteria growing in these two mediums, this one, the bacteria is able to produce growth factors for A and B. The bacteria over here has growth factors capability to produce C and D. Now, if I mix both the bacteria, I'm making them, and then I now grow them in a neutral medium. That means medium which does not contain any of the growth factors. Then what I will obtain is a bacterial colony that has the ability to synthesize the growth factors A, B, C, D. This is a neutral medium or a minimal medium. That means the growth factors are absent. So the bacteria growing over here have the ability to synthesize all the 4 growing factors. Now the possible explanation for this is the bacteria growing over here can synthesize growth factors A, B, A, but cannot grow growth factors C and D. In this case, the bacteria growing over here can synthesize the growth factors C and D, but not A and B. So what happens is, in this particular case, the bacteria is able to synthesize all the 4 factors. So combination is taking place in such a way that the growth factors of the genes, they are combining. That means a new gene with the ability to synthesize the growth factors A, B, and C and D. There might not only be this possible combination, there might be also the possible combinations like lacking in all the factors, but these do not survive since this is a neutral or minimal medium. Only this survive, and this explanation is possible because of recombination. And then combination is possible only because of bacterial reproduction, and in this case the first one was conjugation. By conjugation, what it means is bacteria are coming in contact with each other. Other method of reproduction is by transduction. In transduction, a bacteriophage is involved. Again, this was discovered by Lederberg and Zender. A bacteriophage is a virus that infects bacterial cell only. This is the viral DNA. This is the bacterial cell. So when we talk about bacteriophages, it consists of 2 parts, the head and the tail. The tail connects with the bacterial cell and only the DNA is inserted. Now, in transduction, what happens is this viral DNA can incorporate bacterial DNA. So the bacteriophage that incorporates the viral DNA and the bacterial DNA looks like this. So this is the new viral DNA that contains both the DNA of the virus and the bacteria. It enters the cell. This is the original bacterial DNA. This interferes with the replication of the DNA, and then we get many such new viral DNA, which leads to the formation of protein coats. And again, we get more bacteriophages with the new viral DNA in this bacterial cell, of course, releasing the new bacteriophages. So this is one mechanism of genetic transfer by transduction. In this case, the viral DNA already has a bacterial DNA. This bacterial DNA might be random DNA or some selective DNA. The last method of bacterial reproduction is transformation. In transformation, what happens is free naked DNA enters the bacterial cell and it gets incorporated into the bacterial cell. This bacterial cell replicates, producing more copies of the DNA, and as such, when the bacteria divides due to fission, you get new DNA. Such transformation or this mechanism can be used to explain Griffith's experiment in which he injected mice with two strains of Diplococcus pneumoniae, that is rough strain and the smooth strain. Smooth strain was able to cause pneumonia, rough strain did not cause any pneumonia. Now what happens is, in the last part of the experiment you remember that living rough strain plus heat killed S strain were injected in the mice and the mice still got pneumonia. Now this can be explained with the help of transformation experiment. And again this was further concluded by Avery, McCarthy and Manfield. So these are the mechanisms of gene transfer achieved by reproduction in bacteria. There are three processes: conjugation, transduction and transformation. Now moving on to genetics, there are many important advances in technology. And the most important or useful one is DNA fingerprinting. This technique was discovered by Alec Jeffreys and it has been used to solve crimes or to solve paternity cases. DNA fingerprinting is basically based on repetitive DNA which is unique for each individual. Now, not all of the DNA code for proteins. Some of this DNA do not code for anything, if you remember introns and exons. Now what happens is, repetitive DNA is unique for each individual. Generally in DNA fingerprinting what happens is, this repetitive DNA create different bands. For example, this band belongs to a father, this one belongs to a mother, and obviously the child will have an intermediate pattern. It is because of the child getting some DNA from the father and some DNA from the mother. Now, DNA fingerprinting is based on repetitive DNA which is unique for each individual. Now, how this process works is, any tissue or blood or hair can be collected from the crime scene. Then it is isolated and amplified. By amplified, what means is make more copies of the DNA. This DNA is made to undergo electrophoresis. Because of electrophoresis, this repetitive DNA creates different patterns. This is now transferred to a nylon membrane using southern blotting. Then in the same nylon membrane, radioactive probes are injected. Now, radioactive probes, what they do is they attach to similar repetitive DNA. Alec Jeffreys, what they did was he developed radioactive probes which could attach to the repetitive DNA. And as such, when this nylon membrane is subjected to X-rays, they get patterns of DNA fingerprinting which can be used to solve crimes. gene expression in prokaryotes and eukaryotes are quite different. In prokaryotes, when we were discussing the lac operon, and operon, you basically found out that the genes responsible for producing enzymes responsible for metabolizing lactose controlled by either the promoter gene or the regulator gene. Similarly, in eukaryotes, the gene responsible for producing a protein is known as a structural gene or a producer gene. Each producer gene has a receptor site. Then nearby you will have the integrator gene and the sensor site. Sensor site is activated by signals such as hormones or other molecules. Integrator gene and producer gene produce mRNAs which later code for proteins. Integrated gene works just like the regulator. That means if this mRNA acts on the receptor site, then the producer gene is no longer able to produce the functional proteins. This is how your gene operates in eukaryotes. Human Genome Project was started in 1986 with the help of 17 countries. These 17 countries, what they decided was to find out the entire genome sequence of the human DNA. So, different methods were applied. One was express sequence tags and sequence annotation. I already told you not all of the DNA is useful. Some DNA do not produce any proteins. So in the expressed sequence tags, what they did was they studied only about genes that were able to produce RNAs. This type of method is known as express sequence tags. Whereas in sequence annotation, the entire gene, that is whether a functional one or non-functional one, was studied. So, due to this, quite important information was obtained. The first one is that the entire human genome has more than 3,000 million nucleotides. This entire human genome has more than 3,000 million nucleotides. Gene chromosome 1 has about 2,900 genes, and chromosome Y has only 261 genes. So we can also find out which chromosome has the most number of functional genes. Another important was more than 50% of the genomes or genes do not have any such known function. And on an average, a gene has about 3,000 nucleotide bases. So this is the information that you get from Human Genome Project. Now this Human Genome Project is quite useful in understanding transmission of hereditary diseases. But also there are many ethical, social and legal issues. Like for example, recently in China, there was a proclamation by a doctor who said that he edited genes and created twins. By this what it means is he selected only the useful genes. That means the not so useful genes were not needed. That means these babies would either have very high intelligence, high physical capabilities, etc. So again, this is a ethical issue. Another case is this information can also be used by insurance companies. For example, if a person goes for an insurance and he doesn't know about whether he's going to have a disease at a certain age or not, but the insurance companies, they already have this information, so the person is at a loss. So Human Genome Project is also useful but also quite harmful. So this is the completion of the chapter.